Oh, let me flip this around here. All right, let me mute that. All right. So, see if that'll come up maybe. Thinking about it. All right. All right, so thanks for having me, and I'll try to keep this short. As usual, I have more slides than minutes, so I'll go kind of fast. So I've been getting into this project that I've called Boone's Crawdads, because you may or may not know this, but we are very close to historic trails that Daniel Boone traveled. I got into working on crayfish when Zach Couch convinced me to start working on the Louisville crayfish, which is the end of the Wilderness Road. And so coincidentally, that's sort of where my crayfish journey began, was at the end of the Wilderness Road. So I'd like to welcome you here to Crab Orchard, which is a major stop along the Wilderness Road. I'm going to show lots of maps today. I'm going to show more maps than crayfish today to try and get you hyped about the region and the area that you're sampling in over the next couple of days. So things that I want you to do is admire the faults of the region, and I literally mean the geologic faults and the other things in too. Take in the views. I highly recommend if you're out surveying and you see a scenic overlook, I encourage you to stop and look, because it's likely Daniel Boone and many other explorers stopped at those exact same locations. There's one pretty close by Hall's Gap, which is in Stanford. I went there one time and I thought, man, this was the most open area people had seen in their entire lives when they came to this region. I highly suggest collecting geodes. There's lots of cool geodes around. I'm not saying bring me geodes, but it's a cool thing to look at in the creek while you're finding crayfish. And then I mentioned as part of this project, I'm also getting some video. So I'm going to be floating around the next couple of days, and I'm going to ask you for some of your cray takes. So I've got this little microphone here, and I'm going to ask you what your cray take is. What's your favorite crayfish? What do you like about crayfish? Anything that you have to say about crayfish. Some of you might have seen the Subway takes. It's sort of the same thing as that. So again, my goal here is just to sort of gather footage for this project as I sort of go back through the Wilderness Road, and the Mudbug Mania 2026 is a good opportunity to do that. So I gotta include lots of Daniel Boone quotes from here, and this is the first line from his autobiography, his brief autobiography. Actually, this is not the first line in that, it's somewhere else, but this is on Kentucky. In such a diversity, it was impossible I should be disposed to melancholy. And so I agree, and a shout out to Dr. Schuster. I've stolen all his photos, just like all of you steal his photos for your presentations too. And so credit to him. And so since I've been in this area, I've been really interested in this particular notch here. And so, but before I get into that, I want to talk about things introduced from Kentucky into Kentucky from Pennsylvania. This includes bluegrass, Daniel Boone, and myself. So bluegrass itself, likely European of origin, but they think it came from Lancaster, Pennsylvania. Daniel Boone came from Berks County, Pennsylvania, Reading, Pennsylvania, and I came from Lebanon, which are all three bordering counties to one another in the heart of Pennsylvania Dutch country. Things introduced to Pennsylvania from Kentucky, the rusty crayfish. And so this is one of the reasons that I've continued this project, is because I'm interested in sort of the true identity of the rusty crayfish. But again, I want to give you a little introduction to the landscape. As I was learning about Kentucky when I came here from Arkansas, I was sort of obsessed with this little area here. I couldn't really find much explanation, but it's essentially the intersection between the Knobs ecoregion here, the bluegrass, and the Eastern Highland Rim, essentially. And then we have the Eastern Kentucky mountains here. And so there's this interesting notch here that now I know is formed by the Dix River and the hanging fork of the Dix. But why it's actually sliced through the knobs, why it sort of reaches down a little bit further, I haven't really found a suitable explanation for it, until I started looking at the geologic maps in the landscape. And so I was convinced that this is the most interesting part of Kentucky. So you are here, EKU is here. I live right about here. And so every day I get to drive this drive here, about 40 minutes from my house to EKU, both ways. And so as I do that, I can see off to my right here, if I'm driving to EKU, I can see the knobs, I can see the hills there. And I highly encourage you to sort of do the same thing as you're out in the landscape. Look for the knobs. This was a major geographic feature that people followed. And so if the knobs are on your left, you know you're going this way. If the knobs are on your right, you know you're going this way. And so Boone and many of these early explorers traveled along these geologic features, which are formed from ancient fault lines. All right, and so they didn't realize they were following these faults, but that's essentially the boundary between these. And that's what sort of caused this weird sort of notch in the knobs. This is also where the headwaters of most of the major regions, drainages of Kentucky start. So here we have headwaters of the Cumberland. This is Buck Creek and Fishing Creek here. We have the Green River over here. We have the rolling fork of the Salt and then the main stem of the Salt, which I really treat as sort of two separate drainages because they go a really far away before they meet and dump into the Ohio. And then we have the Kentucky River over here. So it's this really interesting intersection of probably about 80% of the watersheds in Kentucky sort of meet here. And they are divided and have continuously, or through time, been variously joined and separated through this faulting activity in this area. And so just to sort of zoom in on the geology of where we're at, one of the most interesting things I think of this region is these high alluvial deposits. And so high up above rivers are these high terrace gravels, they call them. And I am convinced that these are not from modern day river drainages. They are too high. I'm convinced that these are essentially the ghosts of drainage paths that are sort of stuck up on these elevated places. Now my suspicion, I don't know for sure, but my suspicion is that these gravel sediments are from a river that went all the way through Cumberland Gap. Essentially the river that formed Cumberland Gap eventually went through here. And this area was a major spillway off of the Mississippian Plateau is my hypothesis. But that's sort of a future project. We want to test these gravels and sort of figure out where they came from. And so some of them, I've been sequencing lots of DNA of crayfish around here, and that DNA suggests lots of ancient connections. And so there might still be evidence for these drainages. I actually live here, and it turns out these high fluvial deposits are like right near my house. And so again, these are far too high to be deposited by modern river drainages. So I say this as you're out to sort of look for this type of habitat, because I don't know for sure, but I suspect these might be places where unique burrowing crayfish might be. I don't know for sure. I study the stream-dwelling crayfishes, so you guys are going to do a great job of surveying. So we're going to find these deposits. The other thing that I want you to think about when you're on the landscape is this is really what Kentucky sort of looks like in its cross-section. So it might feel like the bluegrass is sort of lower elevation, that you're going down the elevation, but you're actually pretty high up on the bluegrass. It's this elevated plateau. So the bluegrass itself is almost like this elevated island, and the knobs are almost like a coral reef around it. And it's an interesting way to call it a coral reef because it does contain lots of coral fossils from this Mississippian age. The other thing to think about as you're going on this landscape is it used to be a lot higher than it is right now. So this layer actually laid on top of the bluegrass, all right, and so eventually eroded away. And so I mentioned the geodes. The geodes are in this Mississippian layer. But even though the Mississippian rocks aren't there, the geodes are sort of left over from these historic layers that used to be on top of that bluegrass layer. Just real briefly on the ages of these things, the higher it is, generally the younger the uplift, I guess. It's weird to think about, but eastern Kentucky here, we have Pennsylvanian age rocks. These are sort of your conglomerate, your sandstone conglomerates. That's what you're going to find in far eastern Kentucky. You see Pennsylvanian period, Pennsylvanian, and then we go Mississippian, Devonian, Silurian. So essentially, as you go from here down, you're going back in time. These rocks are younger here in eastern Kentucky. You feel like you're up in the mountains, but it's the uplift that's recent. The rocks are younger, and as you go down, you essentially travel the course between Pennsylvanian and Ordovician, and so you essentially travel a couple of 100 million years through these rocks, through this area. So again, I'm telling you this to sort of give you perspective. I think a lot of this reflects ancient inverted topography, so things that used to be mountaintops became rivers. We have this uplift. Again, you guys are tired, so I won't go through all of these processes, but again, I've sort of just given that idea so that you can think about that as you're in the landscape. So I mentioned the historic trails. This is a map of not just the Wilderness Road, but a variety of other trails, such as the Warrior's Path. And when I was lining some of the DNA up of these, I was like, some of these sort of follow the trails. And I was like, well, this is a great way to merge natural history with human history, pioneer settlement history, and that's been my goal here. So I don't necessarily mean these literally, but I do think some of these might actually be paleo drainages. So here's another map showing where you are. Note Crab Orchard is on these really old maps, so that's the town that you're in. So I mean, this was a really important stop after you got off to the Mississippian Plateau. And so my thinking is that perhaps these are actually ancient river drainages that humans just found the path of least resistance. And they used to be like this, but then over time they eroded and became like this. And so the people are sort of walking on this path, but that path might actually be an ancient river drainage. Those terrace gravels may be the evidence of the ancient drainage. So again, here we have the faults overlaid with the trails. So you notice again, high concentration and intersections of many faults. Some of these lineages, mitochondrial lineages that I look at sort of span some of these. These are all sort of rough approximations. This is sort of a mystery species that we have in the area that I'll talk about in a second. This is Kentucky river crayfish juvenilis, and then the rusty crayfish. And so I think these things might have inhabited really old river drainages that we're unfamiliar with, which would explain their strange disjunct distributions. But they could also be bait bucket introductions, and that's one of the things we're going to try and figure out with some more hardcore genomic stuff. So we're going to ask, was the Wilderness Road an ancient drainage? And are the crayfish older than the mountains themselves, essentially? That would indicate the crayfish have been here since the uplift, or at least the later uplifts of the Miocene, which are about 30 million years ago. So a few of the Faxonius of the Wilderness Road, I'm happy to provide this. I'm definitely going to give it to Eric for the workshop. But we have Faxonius placidus, really easy to identify by spot on its finger. If you find a species with a dentate incisor, it's going to be either Putnami or Christovarius, or we have some really strange intermediates here in the region in orphaned or disconnected streams. So those would be great areas to target. And then if it has a straight incisor and a rusty patch, it's likely your true rusty crayfish. A black wedge is this mystery lineage that I'm following around that you guys will likely find. I'm going to soon stop calling it the mystery lineage, and I think I'm going to call it the Wilderness Road crayfish. I don't know that it's an actual 1 species or 5 species or even just like something else. So I'm hesitant to put a name on it at this point. And if it's neither of those, I generally default to juvenilis. So juvenilis has neither the black wedge nor the rusty patch. And that's about all the Faxonius that you're going to run into in this area. The distribution, I find the Green River, we find Putnami and that mystery. We generally find these pairs. So Kentucky and Dix River, we find juvenilis and Christovarius. Cumberland River is where we find all of them, particularly in Buck Creek. Buck Creek is interesting that they're all in Buck Creek, which again is right over here. The Rolling Fork, we get the mystery, and then the Salt, we get a different one, which is actually really similar to Faxonius ilex, the Moe wild crayfish, which is found below the Falls of the Ohio, which is actually the end of the Wilderness Road and that species I mentioned before that sort of pulled me into this project and sort of made me think of this thread. So I'm going to skip through a bunch of this. There's been lots of interchange between these regions, and I think, again, it's because periodic uplift and erosion have really made the drainages a bit more unrecognizable to us. So I mentioned that mystery rusty. Morphologically, this thing has been called The rusty crayfish, at least based on its gonopore morphology, many of you might know me as a lurker on iNaturalist, where I just look for this one thing and I only favorite this. And so I've been looking for this black wedge and these lateral red spots. And anytime it has that, I flag it, I like it. It doesn't matter what somebody calls it, and a variety of different names of people in this room have called them these things, but I'm just sort of zeroing in on what that one color characteristic. And so I can sort of build a distribution map from my likes, and it falls out pretty similar to what I see in the mitochondrial data. So this is what the main area that I'm sampling here in Kentucky, but it's also popped up in a few places in the Holston that we think is the non-native range, but it might not be. This paper determined that this was something else, Faxonius ronaldi. I don't think it's that. And I think the tools that we used at that time weren't really great for determining whether or not this was introduced there or not. So the idea is that maybe this actually is some sort of cryptic native lineage. This mystery thing breaks out into a whole bunch of different clades. And so again, that's why I don't know if it's one thing or 5 things. And so I'm not rushing to put a name on it, but these lineages all mostly have that black wedge and red spots. So we have this modern exodus. I mentioned rusty crayfish introduced to Pennsylvania in Daniel Boone's own watershed. I'm preaching to the choir here. You guys know about it, but we don't really know fully its native range. And lots of things just get called rusty crayfish, even though it might not actually be the rusty crayfish. So I'm again interested in those areas of North Carolina, which Daniel Boone settled along the Yadkin River. So I've got plans to go out there and sample later on. Buck Creek, Buck Creek is probably one of the most interesting place in this area, in this region, and I think it has served as pathway for multiple paleo drainages, and these lineages are sort of stuck there. They're leftovers of these lineages. And so I'm not going to go into too much detail, but the one thing that is really important is that these disconnected tribs have really unique diversity. So many of these creeks that drain into Buck will go underground before they join the main stem. And at least genetically, it looks like every one could be described as a separate species. It's almost like these are islands, that these things speciated and have sort of gotten stuck there in these tributaries. So as you're out, I encourage you to be in any rinky-dink little stream. Many of these lineages too, in Buck Creek, juvenilis has multiple haplotypes. I think some of them are dry lineages and some are wet lineages. So some of these are really adapt for surviving drying conditions. And so if you go to a creek and it's dry, I would encourage you to still flip up rocks, because you are likely still going to find these Faxonius, these stream-dwelling crayfish, in there. And I think certain haplotypes are more associated with those dry, resistant lineages, and some of the more wet lineages that are a bit more connected. And rusticus, that mystery rusty occurs here too, but it's only in a narrow range. It doesn't seem to be like widespread throughout the drainage. Buck Creek also seems to be the place where the Kentucky River crayfish originated. So Faxonius juvenilis, its oldest haplotypes are here in Buck Creek. And so it seems like this thing went north and maybe went back and forth into so this would be the Dix River where we are here now. And so I think this is the birthplace for juvenilis. Buck Creek shows up all over the tree. These lineages are really diverse. And again, I think there's like little time capsules of ancient river drainages. So I want to thank folks that have done previous Mudbug Manias, because I've sequenced lots of your Faxonius. I've conned my students in molecular biology to sequence lots of them. So it's been this huge data set. We're getting close to being able to have it cleaned up enough to release on GenBank for other folks to use it. And so this is where I'm going to end. So again, admire the faults, take in the views, look at the geodes, and then again, think of your cray takes that you want to give me throughout the week. And so that's where I'll end it there. Thank you very much. Happy to take any questions if you want to get the... Not that one. I know it's late too, and I'll be around. Yeah.
Can you go back to the facts and ice slides? So
sure. Yes, please do. I got the river drainage and I got the color characteristic one there.
Yeah.
All right, there we can do that one there.
Okay, yeah, I saw one.
And I basically put this together because it's, you often don't find Form 1 males. It's not that I don't think that's a good characteristic, but yeah.
You said the mystery thing is also in the clinch?
Holston.
Holston, yeah.
Yeah.
I collected one there that had the phenotype.
Oh yeah, it's in my likes of iNaturalist. I've seen it.
Because I think I called it Putnami.
You called it Putnami, yeah.
That doesn't make sense.
But I wanted to call it out because, I think 2 years before that, you'd also called a Putnami in the Green River that I was like, that's not Putnami, man, And so I wanted to pull them up side by side so I could show you now that you know that the colors are the same. And I think it's because they were in the same historic drainage.
Yeah, I was completely unaware of all of this.
Well, yeah, sure. How would you know? Yeah.
It's like when it's not
Like I
this,
said,
call it this.
I'm just a lurker out there,
Yeah.
and
That's really cool.
So is that Black Wedge mystery guy, is it more related to Rusty's or?
Let me pull up the tree here. The answer is not really that close related to Rusty's. It's pretty far from true Rusticus. And so the Black Wedge here is everything in green, and it falls in multiple clades because it's intergressed with Putnami a bit in some drainages. This is the Green River drainage, this highly divergent clade, and this clade in some iterations is actually more closely related to the Ozarks. Faxonius Robert I, Wagneri, and Eupunctatus. So Green River more closely related to Ozarks. And then this is a clade of, this large clade here I think is the rolling fork of the Salt River, which also has Buck Creek too. Like Buck Creek and Rolling Fork of the Salt for some reason share an identical haplotype. This haplotype is also in the green. So there's the Rolling Fork seems to have gone back and forth. Then I mentioned Elix, which is down here somewhere, I think is in that clade too. And so the rest of the mystery, essentially in the lower green, is sort of split up in these stutters. So you also have the Rough River down here too. It's a nightmare, it's a mess.
Yeah, throwing Christovarius in the mix with it all.
Yeah, and so Christovarius and Putnami are the most basal of the entire group. It seems like these headwater specialists were the first colonizers. And I seem to think that they might have been around during one of the first uplifts of the Cincinnati Arch, which is sort of the big arch that cuts through Kentucky, because the oldest Putnami haplotypes are from the Barren River, and the oldest Christovarius haplotypes are from the Big Sandy. And so I think those would have been connected over that Mississippian layer that laid over the bluegrass, but then that Mississippian layer eroded away, and you end up with Christovarius over here and Putnami over here, is my tinfoil hat theory, anyway.
No, I see it, man.
Yeah.
So you don't believe in bait bucket introductions?
Yeah, I mean, I believe it in sometimes, I mean, I have no doubt Great Lakes, I mean, we've had, we've connected things.
That just always seems to be the answer, right?
I know. So the weird thing, some of those things like the thing in North Carolina and some of the Rusties in the further south, they come out basal to everything to the north. And so that's why I think it might be that those areas are actually older areas because of how they fall out phylogenetically. But just because you go out and barcode it and say, well, it's the same thing as this and we've never found it here before, to me is not good evidence of a bait bucket introduction. But one of the things we hope to do is do something called DART sequencing, basically do some population genetics, and just the idea is that we should see a founder effect, that these places in North Carolina, if they're introduced, should have much lower genetic diversity than say in the native range, or it should be, they both have really high genetic diversity, which you can't tell with this data right now. Yeah.
Does the mystery one have a unique Form 1 gonopod shape or anything?
Everything. That's the difficult thing, so I feel like it's like this ghost lineage that is able to integress with these other ones and sort of, it mingles with this one and mingles with this one, but it's also conditional, like it doesn't mingle with juvenilis, where they co-occur in Buck Creek for some reason, but it seems like historically they might have mingled in Elkhorn Creek in northern Kentucky, which it has the black wedge, but it barcodes as juvenilis. And so, it's the other reason why I'm hesitant to sort of call it a species, because it looks like it might have a completely variable gonopod length across its entire distribution. Yeah. Right.
Have you been able to get some funds together through like genomics or computer viewing?
I'm able to scrape together $3,500 for a dart plate that we're going to send off with the state, because the state is annoyed that they're always getting blamed for these introduced crayfishes. I mean, North Carolina, they're now up to 4 that have come from Kentucky supposedly into North Carolina. So he's going to fund at least the first initial sampling on these wilderness road things, but with the rusty crayfish as sort of his justification. So yeah, Jeff Herod is going to toss a little bit of money. Since I got this, he was like, all right, since you already got the travel, I'll do the plate. So, yeah. And hopefully that leads to more stuff down the road for some other people. Yeah.
So with your Christopher, it is like things. I know, like when I looked at them in like the Upper Tennessee system, some populations will have more standard Braceric and Varus kind of eyes with nice shoulder and one field amounts. But then I come across populations that look more intermediary where their shoulder starts to be Ross.
Yeah.
And the elements are like about half the weight, something which would consider closer to like Ericksonianus or like Spidosis.
Okay.
Do you see the same thing here in the Cumberland system stuff?
I can't say I look at them that much. I really need to get into more character coding and stuff, but simply because a lot of these specimens I don't get to look at first, because they're all the bags from Mudbug Mania, and my students are just pulling off a leg, and they might be females or juveniles and stuff. And so, yeah, that's another reason why I can't say I'm an expert in this group yet, because I haven't gotten there yet. Oh, yeah.
Buck Creek, are they co-occurring in many areas?
They do co-occur in Elrod, which is down further. So yeah, actually the best place to find all. It's my favorite site in Kentucky. I'm hesitant to tell you about it, but it is sort of the meeting place of all these. I think it's right about here, and maybe Davey can correct me, somewhere in this neighborhood. But you find Juvenilis is a wuss there, and the mystery is the big dog there. Juvenilis is the dominant thing in the upper parts of Buck Creek, but the mystery does not occur there. And so Juvenilis is the big dog up here, and they get really large. But where they co-occur, it seems like this mystery dominates it. And then you're also starting to get competition with Placidus and Cumberlandensis, so you get these other bigger crayfish. So Juvenilis really isn't in the lower part at all. I don't think it's a good competitor with the rest of those things. And the mystery thing only occurs in this sort of narrow geologic band. So this is literally like the edge of what do we call it? The escarpment, I guess. And so the mystery thing isn't everywhere. It seems to be tied to the geology of Buck Creek. So. Yeah, but Elrod is the best place. There's a giant scour pool there that I think is like an ancient scour pool that is like moved off the side of the land, a hillside essentially. And it's been an important refugia for these crayfish in these creeks that are really prone to drying out sometimes. Yeah.
Do you ever get anything that looks like a hybrid?
Yeah, so I've been to Elrod about 12 times, and I never once found a rusty patch until last spring. I found one of the mysteries with a rusty patch, and that's it. And so I'm like, well, I barcoded it, and it came out with the mystery. It didn't come out with the true rusty crayfish. So I started thinking about like maybe there is some interesting like genetics with the rusty patch itself. Like it might be maybe some like inbred characteristic or some feature that is recessive or something that only appears sometimes but has become fixed in true rusticus populations but may pop out in some of these relatives like Juvenilis or this mystery thing is my working hypothesis. So right now, again, of everything that's been surveyed here, we only found one individual with a rusty patch. Yeah.
Can you go back to the geology profile of Kentucky?
Sure.
So on this slide, could you point out some landmarks, like where's Buck Creek fall in, where's Louisville?
Well, the difficult part is you can see the transect here. So the transect goes from eastern Kentucky to Lexington down to southeastern Kentucky. So we're not actually in this cross section, but we would essentially be right about, theoretically, somewhere like here. in that Knobs ecoregion, right near the bluegrass, but getting these Mississippian layers there. Again, that's the layer that contains the geodes, which is why it's called to my attention, because I'm like noticing, I'm out catching these crayfish and I'm collecting geodes and stuff. And weirdly, Buck Creek is the biggest geodes that I ever find. Elrod, like this big. Where you go to somewhere like Hall's Gap, I mentioned, is like right at the boundary here. As you go to Stanford and has an outlook, they're like known for these really tiny geodes that have interesting chemical structure. I don't know, but the geodes are really old. It's just I'm noticing this correlation between finding the crayfish and the geodes. So you have Lexington here. We'd be right about here at this intermediate area. And if you go a little bit further east, you're going to get into the eastern Kentucky coal fields. Yeah, Davey.
What about like next door to Buck Creek, the rock castle system, and some of those similar little drifts that sink out and things like that? Is it found anywhere?
I don't know about those sinking tribs, but Rock Castle main stem is boring juvenilis, which is the same juvenilis, some of the same haplotypes we find in Buck Creek. Fishing Creek next door doesn't have any of them except Placidus, as far as I know. You guys might find different ones, but it seems like juvenilis is absent from that. But there really hasn't been much survey data, so I'm excited to see what you guys might find.
Do you have data from Skeg Creek on the Rock Castle system?
No, but one of these trails technically is called Skeggs Trail, so that's interesting.
We'll hit it this week, so.
Cool.
Good for you. Excellent.
Go on.
Okay, one more.
Your big phylogenetic tree,
Uh-huh.
barcodes, right?
Yeah, yeah.
Which barcodes did you use?
Just Co1.
Co-1.
Yeah, yeah. And I keep everything from Gen back in too. I don't delete anything. So I mean, because I think like for a long time people were looking at like, well, this comes out weird, and I think the reason we're coming out weird is because these things historically have intergressed. It doesn't mean it's a continual hybridization process, but I can go far up into eastern Kentucky and get just Christovarius, but some of them will also have juvenilis haplotypes. So it's almost like I will find the mitochondria move upstream into other species. And so I don't necessarily view them as like, the barcodes as species limit indicators. But I think that's why we've had weird problems with barcoding, is because even though the juvenilis mitochondria will move far beyond where the last juvenilis was, essentially. All right.
Thanks a lot, Dave.
No problem.
So for the rest of the orientation part here, I'll let talk about the teams in the watershed, but let's take a 5-10 minute break. Is that good for you guys? And I'll just get my laptop set up, and I might as well show that Excel file and talk about a few items. You definitely must stay for this. No, I'm joking.